We had done what? This topic is going on, or are we done? The first part of the chapter, the sexual reproduction that we studied, DNA copying that we studied. Are you all clear with that?
Yes.
DNA copying clear, all of you? Variation, are you clear?
Yes.
Then the sexual reproduction in different forms of animals that we had studied.
Yes.
Give me two examples which show budding.
Yeast fungus, yeast and hydra. Fungus, rhizopus.
Unicellular organism yeast, and hydra would be the multicellular organism to write. Give an example of a plant which shows budding. Bryophyllum, plant that shows budding. Then next is fragmentation and regeneration, or fragmentation and regeneration. Tell me one major difference. Fragmentation and regeneration. Tell me one major difference. One major difference. Fragmentation and regeneration.
Regeneration, new cells are formed and
No. If it is fragmentation, the body of the organism naturally. If it is fragmentation, body divides into fragments naturally. If it is regeneration, think of the planaria. What did I explain you? If it is accidental, the body divides into fragments, but accidentally. That is not a natural process. What are some organisms that show? Now give me one example each for both the methods, fragmentation and regeneration. Phytophora for fragmentation, regeneration, planaria. What was the function of calyx we understood? We studied no that part? Flower not over. So what was the last topic we studied? Vegetative propagation is over. Vegetative propagation is over. Then today we are going to start with that. Not over. Not over. Okay. So vegetative propagation not too much. Only 3 methods to understand. Vegetative propagation that we understand. Vegetative propagation, we have studied cutting. So now that we are talking about cutting, what are the different methods which part of the plant utilized? Only that to understand. So when we are talking about cutting, which part of the plant is generally used? It is majorly the stem, in certain parts the root. One specific concept to understand for cutting when you get questions, that when we are talking about the process of cutting, say suppose I talk about plant and some stem cuttings has to be done. So now some stem cutting for the process of growing plants. The cut is always done this way. It is always done oblique. How the cutting is always done for the stem, always oblique instead of making the stem cutting straight. So now when we are, yes, already done, yes. So now I would say that the stem cutting would be something like this. The stem cutting is always this way. So what is the significance of the cut? Surface area. Now that it is inside the soil, it should adequate amount of cells exposed to the soil to absorb the water and the rootlets to develop much faster. Some examples, what did we discuss? Which are some plants which show the method of cutting? Rose, sugar cane. So certain few 2-3 examples also to understand. We talk about the next method. The next method is known as layering. This method that we talk about, layering, can be done in certain plants like say suppose the grapes. It can be done for certain type of plants like the jasmine and for the lemon, etc. Certain shrubs majorly. Now here layering can be of two types, natural or artificial also. So natural layering, how is it? If this is a particular plant and then the roots now developing, certain type of plants, now that I talk about any of this, the roots from deep down in the soil start developing some secondary roots and these start running above the soil. What are these roots known as? These roots are known as the adventitious roots. These are known as the adventitious roots. What are adventitious roots? Adventitious roots are actually the secondary roots which develop from the tap roots and now they start running above the soil. Now that they start running above the soil, the points wherever they touch, they would again start giving rise to rootlets. And another second root system develop. And from there, after a particular period of time, the second new generation plant develops. So the same thing again would continue. This will again give rise to another. And there's a cluster of plants generally formed this way. Now for grasses also, one more very common example is the different types of grasses show a specific method of layering this way. So what is happening? The main root starts giving rise to some secondary roots which always happen to come above the soil and run above the soil. And now they touch the soil. Wherever they are touching the soil, they start giving rise to rootlets like they established and a new plant starts developing. So how do we understand that they all are connected? Because whenever you try uproot one of them, quite a number of them starts getting pulled. Whenever you start uprooting one of them, a number of them. So now, if in a series some 10 plants are formed, all of them are identical. All these plants are identical. So these different type of plants, what can be two advantages of layering to write? What will you write? Two advantages of layering. All the plants are identical, plants formed are identical, and large number of plants can be obtained in a short period of time. There is a major difference. There's a major advantage of layering or cutting now, since we are talking about these methods. What is one more advantage, a major advantage of vegetative propagation? Why farmers or breeders would very much depend on this method or would try to go for this method? Cheaper, and they don't have to depend on pollination. There is no chance factor actually. It is definite that these plants will grow by this method and a large number of plants definitely I'm going to get. But the process of growth of a plant by pollination, there is a chance factor there. Then again, the seed is formed. Now that the seed will give rise to a plant or not, again, there's a chance factor. So for every particular type of a plant, for any plant you talk about, if the plant gives rise to 100 flowers, out of 100, only 60 will give rise to the seed, and out of that 60, only 40 will give rise to a fruit. So it is not that every flower gives rise to a fruit. It's not so. So, but then here the chances of giving rise to a new plant is even more as compared to when we are talking about the concept of flower pollination, etc. So this is one concept of layering. But I would say this is a process of natural method. It's a natural process of layering. It is natural layering. Then if this is natural, then what is artificial? Artificial method of layering, again, for certain type of plants would be something, and then the roots coming up. Now again, the process of layering is never for big plants with big trunks. No, it is always for shrubs which has got some comparatively softer branches. So if this is a particular plant, any of these, and then now that the branches develop, so what will be done is certain branches are manually been drifted towards the soil. They are manually been dug towards the soil, drifted towards the soil. And now that they are put towards the soil, they have to be tied with a support, a stick, bamboo, etc. And a heap of soil a number of times are put here. Why all these things? So that this branch, which has been moved dug towards the soil, it should not revert back. So the stick is there, and then the heap, a big pound of soil is been kept here. A heap of soil has been kept here, and a setup something like this. Quite a number of times here, some hormones are also added to speed up the process of growth. Now, what type of hormone generally should be put here to just an offbeat question I'm talking about, which you will generally not be asked with layering, object or cytokinin. Cytokinin speeds up the process of cell division and growth. So now, yes, a variety of antiseptics or disinfectants and different type of disinfectants are there, and then the hormones are used here. So if this is the process of growth being carried out manually, this is what we will talk about as artificial layering. This is what is done, artificial layering for grapes and strawberries and various different type of plants. So layering can be natural, it naturally happens, and it can also be done artificially this way. Common in these two type of plants is methods is plants obtained are all identical. Let us talk about another method which gives rise to identical plants. Again, we are talking about budding. I had already asked you this example. Now, in case of this specific type of plants, now these plants are quite small enough and they give rise to broad, comparatively broad leaves. And these leaves, a special feature of these leaves are that these leaves always result in the formation of small bud-like structures at the margin. They give rise to some bud. When the leaf is already mature, they will give rise to some bud-like structures on the leaf lamina or on the margin of the leaf. Then from this bud-like structure, some fine root hair-like structure develops. Some fine root hair-like structure develops. Which plant am I talking about? I am talking about plants like Bryophyllum. I am talking about plants like Bryophyllum. Now, these leaves, what will happen is that quite a number of times, these leaves, which has already given rise to this fine hair-like structure, they start touching the soil and penetrating. Quite a number of times, this leaf must have fallen on the soil, and then these buds and the fine hair-like structures come in contact and they start penetrating. Either of the methods generally happen. So, in any of the methods, the hair or the root hair-like structures, they are more or less establishing. After some time, definitely the leaf will all get decomposed. By that time the roots have, yes, my darling, please look here. Nothing is written on her face. Everything is here now. So now, when we are talking about this, these fine root hairs, they will touch the soil, penetrate, start establishing. So by the time the leaf is all decomposed, and from here, we get new plants developing. We get new plants developing. If this is what we are talking about, this is also known as budding. A specialized method of growing new plants, large numbers, but this is only for specific few plants in case of bryophyllum, where the plant is very short, but the characteristic formation of buds in the leaf is a major feature that is found. Common in all these three methods, again remember, giving identical varieties of plant, identical types of plants. Now, an exceptional method when we are talking about vegetative propagation is, we are talking about grafting. Now, this is very, very important method, and you get questions on this also. What is grafting? Grafting is a method of vegetative propagation by which we can obtain improved varieties. Grafting is a method of vegetative propagation which gives improved varieties. Let's see how. So when we are talking about the concept of grafting, I would see that here, which type of plant parts are used? Stem would be used, roots, buds. So these are certain parts of the plant that can be used in the entire process of grafting. What is done? In this process of grafting, we talk about two plants, say suppose. We have 2, 3, 4 different type of plants also can be taken. Related species they have to be. So I talk about one type of a plant, and now maybe I talk about that this is one specific type of a guava, specific type of a rice, any type of a plant that we understand. Now this is one type of a plant. It has to be similar. The type of the plant has to be similar. So I'm talking about two plants here. This is one variety of mango. This is the other variety of mango. For example, this particular variety has certain characteristics. For example, fruits are nutritive, but they are extensively sensitive to pests. These are typical plant characteristics actually. I'm talking about one particular type of a crop, one particular type of a fruit. Every particular type of a fruit has got a list of some advantageous characteristics and a list of some disadvantageous characteristics also. So here I'm talking about one very good characteristic, and here I'm talking about not a very good characteristic. The plant is sensitive to pests. That means very fast the plant will start developing some different type of infection in the fruit. So definitely the farmer is not able to market anything here. What about the other side? Here, yes, what about the fruits? Are the fruits nutritive? Okay, the fruits are also comparatively nutritive. What about this particular feature? No, these plants are resistant to pests. They are resistant to pests. They are resistant to the excessive amount of water in the soil, salinity in the soil, so many factors. Now, to just give you a fair idea here why the concept of grafting is done. The farmer generally compares 2, 3 types of plants of the same type. It can be roses, it can be cauliflower, it can be guava, it can be mango. So what he does is he gets to see that in these 3 plants, one plant has 7 good characteristics, the other plant has 5, and the other plant has 2. But he wants to bring all these 3 characteristics in one, and he wants to do away with this characteristic. He wants to do away with this characteristic, and he wants to bring this and this characteristic together in the same plant, so that can be a productive plant for the breeder. This method requires expertise. It is generally tried. It's a method which has to be tried by specific scientists, biologists. It is not done by the farmers, remember. the testing is not done by the farmers. So now what are they doing is they will keep a plant which has got a large number of characteristics stuck to the soil. Now this will be the main support of the plant. The stem is kept here. Now this particular region of the stem is known as the stalk. The stem which is acting as a support. Then what about this one? They will cut out a small portion of the stem and put it here. Say suppose I understand that this is the stem which is being brought here. And after that what? Again with the help of some tissue tapes. Now these are some specific agricultural methods I'm cutting short. Now these are these two tissues when they have been kept together, 2 or 3 been kept together, they will be tied up with some tissues, tapes, ropes, etc., allowing only the two portion of the stem to remain together for a particular period of time. Allowing the stem and the root, the stem and the bulb, the stem and the buds of 2-3 different plants stay together. I'll come to you. Let me finish first. So now that we are talking about this part of the stem, this part of the stem. This part of the stem attached to the soil, stalk, and this known as the sheath. And then, I'm just talking about 2 right now. It can be 3, it can be 4 again. If there are any questions, remember, it can be 2, 3, 4 also. But what is to be taken care? Can I talk about grafting between the mango and the guava? No. Breeds of mango, breeds of guava, like that. Now this is there, and similarly here what has to be applied so that there is no infection? Disinfectants. Disinfectants to be applied so that there is no infection, because 2 different type of tissues coming together, possible there will be some bacterial fungal infection. So for that use this. Then next is what? Apply hormones, or everything is there. In a period of some few weeks, it is observed that yes, they have started giving rise now to new tissues. They have started giving rise to new leaves. And the stem has now started growing. Part of them will dry out, fall off, and a new green stem has started growing up. The older stem is not reviving. Remember, the older stem will die out. This particular stem will die out. But a new stem starts growing now. A green tissue starts growing now. The leaf starts growing now. The flower starts blooming. So this particular plant, which is a hybrid now, but method is what? Grafting. What is the method? Grafting. And now that it gives rise to the fruit, the flower, the fruit, it is found that this fruit has, this part or this plant has this feature also of the fruit, and this feature also there in the plant. But will it happen at just one go? No, it is just trial and error. So many methods, so many processes. But are they able to obtain such type of developed plants by grafting today? So many. So many. Quite a number of these crops and cereals and grains that we are consuming today are all the hybrids or the grafted varieties. So many of them. So that's the reason a breeder always wants, why not 5 special features in one particular plant? So now that particular plant, they keep on growing and growing and growing. Yes, tell me.
Mama, I have a doubt. Stop. I didn't get what to stop.
The part of the stem, one stem has to remain stuck to the soil. That support plant. So the support plant is known as a stalk, and any type of a tissue that you bring from outside, other different plants that you bring, that will be termed as a sheath. Correct? And then this is physical with the help of some tapes and the ropes and variety of this and that. These are typical agricultural methods that I told you. And then new tissues do develop by this method, and improved plant will come out. If that is the method we are talking about, this is what is grafting. Significance you are always asked. What is the significance? Advantage of grafting is what? Advantage of grafting is they give rise to improved varieties. And what about these 3 methods? Identical varieties. The same variety they will give rise to generation after generation. What will that give rise to? Always an improved. Every time an improved variety. Grafting, always an improved variety. Are these 4 methods clear now? And few examples to understand the differences to remember. What is the significance of vegetative propagation? That also I told you just now. Significance is always, it is more a surer method. There is no dependence on any agents. The process is not dependent on the wind, on the water, or any other type of biological agent also. And it can be manually monitored also. Next, we start understanding the process of, after the concept of vegetative propagation, we study the sexual reproduction. We start with the flowers now. A concept of sexual reproduction in flowers. In flowering plants, what are these flowering plants known as? Angiosperms. When you come across this word, not to get confused, only that we are talking about flowering plants. We are talking about flowering plants. What are angiosperms? Flowering plants. In this type of plants, one specific feature to understand is that a specific part of the flower, a specific part of the plant is known as a flower. Then how to define this word? What is flower? What is a flower? What is a flower? Those of you who have finished this chapter, you should know, no? Some of you have finished this chapter, no, in school, you said. Then what is the definition of flower? That's the first thing that you should learn here. What is the definition of a flower? A modified shoot which contains the reproductive organs of the plant. A modified shoot which contains reproductive organs. It is a modified shoot that contains the reproductive organs. Which are the reproductive organs in case of the flower and how the process? Let us try understanding. First of all, we'll talk about the basic structure of a flower. All of you will have to draw this diagram. Very simple a diagram. Take out your pencils and very neatly we'll draw this, half a page only. Neat label. As I explained, all of you should finish drawing the diagram. I'm not going to give you any extra time for this. It's very simple. Who is talking? I asked you to draw, no? For drawing also your mouth has to work so much. Now what is this diagram about? You will be asked to draw this diagram. Very simple, easy to pick up those 2 marks, 3 marks. What to do? Just a presentable diagram to draw, neat pencil, no color shading, sketch pen, crayon, nothing to be used. What is this diagram? This is the longitudinal section of a flower. The longitudinal section of a flower. What are the parts? Let us learn what are the different parts of the flower. The parts of the flower, what are they termed as? The parts of the flower are known as holes of the flower. What are the holes? WH. W is silent here. Now H, the pronunciation starts from H. The holes of the flower. Now what are holes of the flower? The parts of the flower. What are the parts? The first one we start with is the calyx, also known as what? The sepal. The calyx, also known as the sepal. Which one here? We talk about the calyx or the sepal, generally green in color, generally 5 in number. Now this sepal or the calyx, what would be the function here? The calyx or the sepal, they are basically green. I already mentioned that these are green in color, leaf-like. These are leaf-like. They are not leaves. When we are talking about leaf-like and leaf with the veins and everything, no, they don't have those vein structures, etc. What are the functions? Number one, the function is they protect the flower bud. Protection is one major function for them. They protect the flower bud, and next is, since they are green in color, they also undergo the process of photosynthesis. They are green in color, they also undergo the process of photosynthesis. What are the two functions of calyx? Also to write, sepal also to write. Carry out the process of photosynthesis, always to mention. Next, we talk about the second hole. What is the second hole known as? The second hole is known as the petals, or also known as the corolla. Petals or corolla. Function is what? We talk about the petals. What are some features? Features, first of all, what to know? They are brightly colored. Yes or no? Petals are always brightly colored. They are showy. They may have fragrance. But for what good reason all these features? Insects for pollination. So that's the function that you write. What is it? Petals, what are they meant for? Attract agents for pollination. That is the function. Attract agents for pollination. We'll have to also understand what is the basic concept of pollination. Attract agents for pollination. Let us talk about the other side. We now talk about this part. What is this part known as? The anther. What is the job of the anther? The job of the anther is to produce the pollen grains. Job of anther is to produce pollen grains. What about the filament? This part is known as the filament. What would the filament do? The job of the filament is to support. What is the filament doing? The filament is only helping in support. The function of filament only support the anther. Produce pollen grains. And here we talk about support the anther. So now together the anther and the filament, what do they give rise to? The anther and the filament together result in the formation of the stamen. What is stamen? I just now mentioned. What is stamen? The male reproductive part. Stamen is the male reproductive part of the flower. Name the parts of the stamen. What to write? Anther and the filament. And the job of the anther I just now mentioned. Now we talk about the female part. This is known as the stigma. And this is this funnel neck shaped structure known as the style. And this is known as the ovary. So when we are talking about the stigma, style and the ovary, let us talk about the three parts that the female part has. So we are talking about the stigma, style and ovary. And what is this collectively known as? The carpel or the pistil. The carpel or the pistil. So what are the four holes again? I talk about the stamen. And what is the stamen made up of? It is made up of the anther and the filament. Talking about the fourth part, that is the carpel. The fourth hole, that is the carpel, also known as the pistil. What is it made up of? It is made up of the stigma. What is the major function of the stigma? To remember, it receives the pollen. Function of the stigma, it attaches to the pollen grains, receives the pollen. Second is style. What is the function of the style? Provides nourishment. The style provides nourishment for the germination of the pollen grain. We'll learn what is meant by germination. For the germination of pollen grain. Lastly, to remember, what is the function of the ovary? What is the function of the ovary? It is the site of fertilization, and this is a very important point to remember. It is the site of fertilization in case of a flower. It is the site of fertilization in case of a flower. A very important point to remember, and forms the fruit. And forms the fruit. So now when we are talking about the 4 different holes of the flower and their function should be very clear. What are the 4 holes? Calyx. Functions, the number and the color. These are only the few features that we are studying right now. The corolla and the petals, what for? What is their function? Attracting the agents for pollination. Stamen, what are the parts? What are the functions? Majorly, we are talking about the function of the anther here, producing the pollen grain. Carpel, 3 major parts of the carpel pistil is the stigma, the style, and the ovary. Once you are acquainted with the parts and their function, now what are the chambers of the ovary? Now, this is not exactly here in the structure. You may not label also. If you don't label also, it's okay. But you should know in the next diagram, we are going to label this. The chamber of the ovary. What is the chamber of the ovary? Known as ovule, and it is inside the ovule that the process of fertilization takes place. Any doubt till here? The structure of the flower is clear. The parts and the functions clear. So this parts and the functions as it is, you have to remember. Now understanding is the next topic, where you have to understand what are the steps and certain questions related answer. Now, with this same diagram, we proceed on to understand how exactly the flower gets transformed to fruit. How exactly the flower gets transformed to fruit, and this process is known as what? This process is known as double fertilization. What is this process known as? This is known as double fertilization. What is double fertilization? We'll have to define first of all. What is double fertilization? Try understanding. This is the ovule. Now in this next topic, we are going to understand this. How fertilization takes place here in the ovule. Ovule is the region of the ovary where the process of fertilization will happen. But now, why the word double? The word double because 2 sets of fusion are taking place at the same time, inside the same ovule. Hence, the process double fertilization. Are you done with all these points writing down? Make it fast. Make it fast. Clear? Basic to understand calyx, what it does, corolla, what it does, stamen, what it does, ovary style, stigma, what it does. So these functions first of all to understand. Once you are done, we'll have to take the definition of double fertilization. Write down the definition of double fertilization. We try to learn the steps. What are the steps of double fertilization? Let us start with that. What are the steps of double fertilization? First of all, define the process in which double fertilization, the process in which fusion of gametes takes place twice. Process in which fusion of gametes takes place twice in the same ovule. In the same ovule is known as double fertilization. I repeat, the process in which fusion of gametes takes place in the same ovule twice is known as double fertilization. We are going to understand twice that we are talking about, why that concept twice. But before that, Fertilization is very much dependent on another process. What is it? Pollination. Fertilization is very much dependent on another process, and that process is known as pollination. So now, what is the definition of pollination and what are its types? What are its agents? So when we are talking about the process of pollination, say suppose I talk about this is the process of pollination taking place in certain flowers. So pollination is happening. Now what is the definition of pollination? Who knows? From the stigma. So that's a standard definition, you write. What is the definition of pollination? Transfer of the pollen grain from the anther to the stigma. Now that we are talking about the transfer of the pollen grain from the anther to the stigma, it may be something like this. Within the same flower. Or it may be flowers of two different flowers, but of the same plant. Two different flowers of the same plant. That is also possible. Or it may be two flowers of two different plants. Now, which one is self and which one is, you already know the words, but what is now known as self-pollination and what is known as cross-pollination? Which one is self? Self-pollination would be as long as the plant is safe, it is always self-pollination. As long as the plant is safe. So then this is also self-pollination and this is also self-pollination. Then which one is cross-pollination? So now I say the processes A and B, self-pollination, and the process C only is cross-pollination. So what is the difference now? The difference is when the transfer of the pollen from the anther to the stigma involves two flowers of two plants. But again of related species they have to. Then the concept is cross-pollination. Very quick we will write these differences. Write down. Difference between self and cross-pollination. Difference between self and cross-pollination. Difference between self and cross-pollination. Write down. Under self, we will write the transfer of pollen grain. Under self, transfer of pollen grain from anther to stigma. I'm sorry, from anther of one flower. From anther of one flower to the stigma of the same. Anther of one flower to stigma of the same, or two flowers of the same plant. Or two flowers of the same plant under cross you write transfer of pollen grain. Under cross you will write transfer of the pollen grain from anther of one flower to stigma of another flower of another plant. Stigma of another flower of another plant is known as cross-pollination. Second difference. Second difference under self-pollination always gives rise to identical varieties of plants. Always gives rise to identical varieties of plants. Under cross-pollination you will write gives rise to improved varieties of plants. Under cross-pollination, you will write gives rise to improved varieties of plants. Now, let us try understanding what are the steps of the double fertilization. What are some of the steps of double fertilization and why the process is as such called? The concept is double fertilization. Why do we term them as such? Now, in this case, when we are talking about the concept of double fertilization, here we understand that the gametes, first of all, we have to start with the male gamete. We were already talking about the anther. Let me start from there. I talk about the anther. What was the job of the anther? Produce pollen grains. So now from this anther, I would understand, yes, the pollen grains have to undergo a process known as germination and the male gamete. So that is what first of all we have to understand. How? This is the male gamete. One single this is the pollen grain, one single nucleus, and this pollen grain will undergo various stages of growth, ultimately resulting in a structure something like this, and where the nucleus now starts dividing into two parts. What are these known as? This part is known as the generative nucleus. And what would the other part known as? What is this known as? This is known as the vegetative or tube nucleus. What is the next stage then? The generative nucleus will again result in the formation of two more nuclei. That would be termed as the male gamete. So this stages for understanding so that you know how exactly the gametes are formed in the flower. So now I would understand how many gametes ultimately? Two gametes. What about this vegetative nucleus, source of nourishment? Once the male gametes are formed, they are absorbed. They are nowhere to be seen. They are not there. I'll break all your bones that you have in your body. Are you understanding? You have seen the sweeter side of me. You haven't. Thank God. If you are not in a position to see the other side of me, better finish your course and go very nicely away. Very nicely to sit in the classroom. Hands are becoming too long. Refill over होने के लिए कौन हंसता है? Refill खत्म हो गया. Allow the refill. Refill चाहिए. Now, when we are talking about this, understand the generative nucleus will give rise to two male gametes. So, how many male gametes per pollen grain? Two male gametes. Try understanding. So, now here we are talking about male gamete. These are known as male gametes. Parallelly, if I have to understand, then this particular pollen grain, now that it comes in contact with the stigma, what did we write? Function of stigma is what? Pollen grains during the process of pollination. And now that this particular pollen grain receives, what was the function of the style?
Provide
Nourishment during the process of germination of the pollen grain. ये जो पूरा process है, this is only one or two diagrams that we draw now. There is a whole lot of process involved, one by one, the process of development of the nucleus and then the germ tube and then the pollen tube. So here I would say that this pollen tube is developing and resulting in the formation of a long tube-like structure. But what is the process involved? It is nothing but continuous cell division. And where now the gametes, the two male gametes here. The pollen tube is nothing but a cellular structure, and for the development of cells, the nutrients which are all packed up here is utilized. So to understand it this way, what about the female gametes? Now these are the male gametes we know. What about the female gametes then? Where are the female gametes formed? If I have to talk about the female gametes, I straight away have to think about the ovule. What is ovule? I told you. What is ovule? The chamber of the ovary. Be very specific. What is ovule? Define ovule. Name this part. Define the structure. This is a ovule that we understand. This is a ovule that we understand. Is it that every ovary has only one ovule? No. The ovary may have 6 ovule also, 20 also, multiple also. Now see, if I talk about certain fruits, we get to see, no, there are many seeds. Many seeds, what impression? The impression is that this particular flower must be having many ovule. At the same time, certain fruits we come across only one seed. So here, for understanding, we are taking only one. Now, this ovule will undergo a process of cell division, meiosis. I just mentioned this process, not necessary for you. This ovule undergoes a process of cell division, and since the process of cell division is elaborate, more elaborate than this, so the number of gametes are also too many. How many gametes in the ovule? How many gametes in the ovule? 8, 2, 6, and 6, no? 6 up there, 3, and then 8 there. So what are the name of the gametes? That also you'll have to remember, and their location. Let us write it down here. What are these gametes known as? These gametes are known as, these are the female gametes that I'm labeling. These gametes are known as the antipodals. 3 at the anterior end, and then 2 at the center. These are known as the polar nuclei. A big gamete at the center, and this is the potential gamete in case of a flower, which generally undergoes fertilization. So what is this gamete known as? This is known as the egg cell. Why I mark this? Because this is the one which is going to undergo the process of fertilization, and 2 more gametes on the lateral sides known as synergids. Done. So the female, that is the ovule. How many gametes? 8 gametes. Name of these gametes? Antipodals, 2 polar nuclei, egg cell, and 2 synergids. Remember the names of the gametes. How many types? 4 types of gametes we have been talking about right now. The rest of the other parts, nothing much to understand. The style, stigma, ovary, you already know. Except for there is a small opening here through which the pollen tube will deliver the gamete. And what is this known as? This is known as micropyle. The steps of double fertilization, first to understand what are the steps. The first step that we understand would be fertilization. Now, all of you, I want you all to write down the definition of fertilization, because you often get confused with the next step. What is the definition of fertilization? Fusion of one male gamete, and one female gamete to form zygote. That is what is known as fertilization, and always to remember. So what is the first step is the first male gamete gets into the ovule. All of you look into the figure. What is the first step of double fertilization then? The first step is this male gamete will enter by the way of the micropyle and come in contact with the egg cell. What is this process? One male gamete, one female gamete fusion, always fertilization. What should it give rise to immediately then? Immediately it will give rise to a zygote. That is the definition already we had written down. So what is the step of fertilization? The first male gamete fuses with egg cell. The first male gamete fuses with the egg cell, and what does it give rise to? It gives rise to the zygote. But the zygote will immediately enter into a process of cell division and result in the formation of embryo. Then what is the difference between the zygote and the embryo? The embryo is a multicellular structure and the zygote is a single cell. The zygote is a single cell. Am I understood? Are you all following? So now, what is the first step I'm talking about? Fertilization. What is the first step happening here? First male gamete enters, fuses with the egg cell and results in the formation of a single cell structure. So what is this single cell structure now known as? What are we going to name this as? This we cannot name it as the egg cell, rather we say this is the zygote formed. So by the time this process happens, these two polar nuclei will fuse together. So let me take this as the second step. So I will say now next what happens? Fusion of two polar nuclei. What will it give rise to? This process gives rise to a secondary nucleus. Can I say this process is also fertilization? Fusion of the two polar nuclei now giving rise to a single cell, and I am saying that this process is now resulting in the formation of a secondary nucleus. Is this process also fertilization?
Yes.
Think what I am saying and understand what I am saying. पहले जो एक मेल गैमेट और एक सेल का फ्यूजन हुआ था उसको हमने फर्टिलाइजेशन डेफिनेटली बोला था. Why? Because it falls in the definition, one male, one female, two from the zygote. But now the two polar nuclei fusing together to form the secondary nucleus, fertilization है? क्यों नहीं है? Both are female gametes. So fusion of two polar nuclei to form the secondary nucleus is definitely not fertilization. इसको fertilization हम नहीं बोलेंगे. Now next is what? This male gamete now gets in and it will again fuse with the secondary nucleus, resulting in the formation of another structure, a single cell but made up of three gametes. Two polar nuclei, एक male gamete. So this secondary nucleus now fuses with what? The second male gamete. Fuses with the second male gamete. And what does it give rise to? This will give rise to the endosperm nucleus. The endosperm nucleus. It gives rise to the endosperm nucleus. So जैसे ये fusion हो जाएगा, now we will not call it as the secondary nucleus, but इसको क्या बोलेंगे? This is known as the endosperm. अभी now this is the deciding point in a ovule. अब ये endosperm grow होगा कि ये embryo grow होगा? Now this is already, by the time this endosperm is formed, this has already resulted in the formation of the embryo. So now when these two structures are there parallelly, एक embryo है, यानी are you following or not? एक embryo है and one endosperm is there. Now, are these both these structures now growing together and developing together? नहीं, only one will develop. या तो endosperm develop होगा या तो embryo develop होगा. दोनों में से एक ही develop होता है. Now that depends. कौन से तरह के seed we are talking about? Which type of plant we are talking about? अगर dicot plant है या तो monocot plant है, I may understand that am I talking about plants like the pea and the beets, or I am talking about plants like the maize and etc. So plants dicot होता है. I'll take just 5 more minutes. Hold on to your patience. Now when we are talking about the dicot plants, monocot plants, अब कौन सा structure develop होगा depends on which type of a plant. According to your syllabus, we are studying this most commonly occurring plants. तो अभी इसमें क्या होगा? The embryo will start developing much faster and the endosperm will not develop. I talk about a PCA. Cotyledons होता है. Gram उसमें cotyledons होती है. Is the gram and the pea the same like the maize? No, absolutely structure form different है. So maize एक monocot है जिसमें endosperm develop होती है. Pea, gram उसमें embryo develop होता है. So let us take this example here as per your syllabus. So after that, what is going to happen? This embryo now, if it is this type of a seed, embryo starts developing and utilizes the nourishment packed up in all this. This is cytoplasm. Cytoplasm always packed with nourishment. तो ये एंडोस्पर्म, एंटीपोडल, सिनर्जिड सब यूटिलाइज हो जाता है in the repeated cycles of cell division. So all these are utilized, and ultimately a structure something like this is all built up. And what is this now? This the multicellular structure that is got that is developed here by repetitive cell division. What will we call this now? As the embryo got transformed into a seed now. The embryo got transformed. The embryo from that small zygote to form a big embryo, a number of cell division had happened. Cycles and cycles of cell division. Which type of cells provided nourishment? The antipodals, the endosperm, and also the synergies. And after that, all these parts will dry up and fall off. All these will dry up and fall off, and this will entirely take the structure of a fruit. This will entirely take the structure of a fruit. What are the parts now? This I will say, this is the fruit wall, which is also known as the pericarp. This is the edible part, the cotyledon, if I am talking about, or any other fruit like the mango, guava, etc. that we are talking about. What about this region? This is known as the seed coat. And what about this particular region? This is known as the seed. Now tell me, this seed is it the ovule part which got transformed into the seed? It is the ovule, that was the ovule inside. So which part got transformed into the seed? The ovule. And what about this entire region? This was the ovary, and this entire ovary got developed into the fruit. So the process of double fertilization, just one more thing I have to tell you. What is this particular process known as? Take a note, this particular process is known as triple fusion. Okay? What is triple fusion? The second phase of fusion jo hota hai, that is called triple fusion, and the first concept is just fertilization. एक बार जाओ घर पे जाके एक बार फिर से पढ़ो in the volume all the steps are given very nicely. Anything that you don't understand, ask me again next class.